Hello and welcome to Medical Dialogues Journal Club. I am Dr. Nandita Mohan, and today I will be discussing a very interesting article that was recently published in the Asian Journal of Oncology, which is published by Scientific Scholar. I will be talking about the action of ultra diluted ethanol extract of Bryonia alba on the HepG2 liver cancer cells. Now, we all know that alternative medicine comprises of well-known ancient Eastern practices of acupuncture and even Tai-chi to herbal medicine, chiropractic manipulation, Reiki, aromatherapy, massage therapy, folk healing, to name a few. And all these are generally used interchangeably with the word alternative medicine, a designation which was created in the 19th century that distinguished these practices as an alternative to allopathic medicine. Now, in this study that I'll be discussing today, I'm going to talk about how the authors explored Bryonia alba as a new anti-cancer agent against the liver cancer cells. So, to talk to us in detail about this research, we have with us today one of the authors, Dr. Satadal, from the Department of Tissue Culture Unit, Heritage Institute of Technology, Chowbhaga, Kolkata, India. Welcome to Medical Dialogue, sir. We are really happy to have you on board here with us today.
Thank you very much.
Great. So, firstly, thank you for agreeing to discuss your recently published article. That is, we're talking about the action of ultra diluted ethanol extract of Bryonia alba on the HepG2 liver cancer cells. Thank you for joining us and giving us your valuable insights on this interesting research that you've recently done. So, firstly, sir, if you could just give us a brief about how was your experience as an author writing this engaging article and subsequently getting it published in the journal. How was your experience?
So actually, it was a very, very important experience, good experience. The process of publishing the manuscript was very simple and smooth, and really we are thankful to the editor of the Asian Journal of Oncology for considering our research article. And the guidelines for the authors was described very vividly, and those are very simple to understand and follow. The time taken for the important decisions, unlike the peer review, acceptance of the manuscript, etc., were quite reasonable. And overall, the experience is very great.
Great. Thank you so much, sir, for sharing your experience. Now that we're talking about this study, if you could just give us a brief about the rationale or the reason behind this and why and your team conducted this actual research and its overall clinical significance. If you could also highlight on the key takeaways for fellow clinicians or researchers in this clinical domain and also for patient education. Are there any key takeaways that you would want to highlight on?
Actually, this study was performed in the Heritage Institute of Technology in the city of Kolkata, and that is our first aim is the selection of the study, the study title, and so we selected the liver cancer study. It is because the incidence of the liver cancer is very high. So in male it is approximately the 7 per lakh population, and in the female it is approximately 1 to 2 per lakh population. So it is very high, and similarly the mortality that is also very high. In male it is approximately 7 to 8 per lakh population, and in female it is approximately 5 per lakh population. And the ratio in between the male and female is approximately 4:1. So the male suffers more in comparison to the female. And if we consider the incidence in India in 2022, it was approximately 30,000 the male share affected, and approximately 11,000 the female share affected. So the number of patients is quite high. We selected Bryonia for this study, and it is because it is an alternative medicine, and it is used in the practice of homeopathy. And some symptoms of the liver cancer are common in which and this particular medicine is applied. So we selected this medicine, and we selected one particular strength of that medicine, and that is the 6 years. So it is a very diluted form of that medicine, but still some chemicals are present in this medicine. We selected HepG2 cell line, and it is because it is a liver cancer cell line, and it was originally isolated from a male patient, 15 years old, and this particular cell line is very important because we can study the different drug metabolism as well as the liver cell toxicity in this particular cell line. We also and this particular cell line is also important because and the persons working with this cell line will not be affected, so it is not tumorigenic, and that is why this cell line is important and is related to this cell line. Now there were different steps in our study. So the first we procured the cell line, and then we standardize it, and we maintain it properly, and then we applied and this Bryonia alba on it. Now the first observation was the changes of the morphology of the cells. So we find that the cells become very small in size, and also there are the degeneration, and there are the destruction of the cells. Now after that we studied the cytokines, the different cytokines, particularly the inflammatory and anti-inflammatory cytokines. So we observed that and these cytokines, the cytokine genes were suppressed, but inflammatory genes and they still they were more in comparison to the anti-inflammatory gene action. So and due to their high activity, the cancer cells were damaged and they were destroyed. We also studied the viability study of the cells, and also the toxicity study by MTT assay, and also the DNA fragmentation. We observed that the DNA fragmentation was more after application of the Bryonia alba. So in short, these are our findings.
Great. Thank you for giving us a crisp and a concise version of your research. Definitely, it's an interesting one. So, based on the findings you just described, the detail and also the conclusion that was drawn, what will be the next steps for you and your team in terms of research as well as in clinical implementation aspect?
Actually, after this, we shall study this experiment in animal model. That is particularly the mice, and then the human trials will be done. And the human trials, there are different phases. So the phase 1, phase 2, and the phase 3, with a different number of the number of patients and the different aim. Now, the first of all, we should take permission from the ethical committee of our institute and the particular hospital from which the human volunteers the suffering from the liver cancer will be taken. Then after getting the ethical committee permission, the one registration should be done with the central government registry, and after that we can start the experiment, particularly in the human trial. And after completion of these trials, and this should be applied in the different patients for the treatment.
Good to know that, sir. So lastly, if you could just lay out your experience as an author, which interacting with the editorial team of Scientific Scholar, and also subsequently getting your findings to present your findings to a larger audience through Medical Dialogues. How was your experience? If you could just share a little bit on that.
Actually, this was a great opportunity for us, and we are really thankful to the authorities of the journal and publishers and for considering our article and allowing us to represent this and to the larger intellectual audience. And we also thank you all for considering us in this particular interview.
Great. Thank you so much, Dr. Das, for joining us today and also giving us your valuable insights on this article. And it was lovely to be interacting with you. Thank you for joining us.
Okay, thank you. Thank you very much.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.